All right, and away we go. This is it, Tuesday, December 7th, 2021. This is the last time you have to see me if you choose. So it is our last lecture. We are going to do a really brief introduction to human development, and that shouldn't take the whole class time. So we should have plenty of time for a question and answer review, an extensive review covering everything, our last remaining lab and lecture exam, and any questions you may have on the final exam. And that's it. Then we're done. Then it's all tests all the time. And 1 week from today, you are finished and you can finally get to your Christmas shopping. All right. Any questions before we get started? All right. Then let's pick up where we left off. As I mentioned, there was a lot of vocabulary in this, and we talked about that in our last class. We did that, so I won't go over that again. And as I also mentioned, we are going to focus primarily on the beginning of this process, kind of picking up where we left off with all of the processes involved in the fertilization of that egg to form that zygote, and then basically all the events that occur, or at least most of the events that occur, leading up to implantation. Then, as I mentioned, we'll pretty much fast forward to the end of the process and talk about parturition and what happens after that. So that is going to be our goal and our game plan for the day. And again, it's going to be a real broad overview with a lot of hand waving because obviously, as I said, we could spend a whole semester talking about these topics. Again, our goal of the reproductive system is basically to take our two haploid gametes and merge them together into a single cell that would have 46 chromosomes. And as we've talked about, that single cell, once that egg is fertilized, as we know, becomes a zygote. Now, after copulation occurs and the sperm is presented to the vaginal canal, it only has to travel about 5 inches. It isn't necessarily a long journey that it has to take, but it is an incredibly treacherous journey. It can actually happen relatively fast, in as little as 3 minutes for the sperm to travel that distance, but it also can take several hours for the sperm to get there as well. And as we talked about, there is a massive amount of attrition that takes place. Remember, we talked about how a small level of sperm would be 200 million sperm that enter into the vagina. Of those that entered into the vaginal canal, if that was the amount that entered into there, only 1% of them would actually reach the head of the cervix, where that cervical canal is located. Of those 2 million, only about 10,000 would reach the uterine tube. Now, if you think about this, we do have 2 uterine tubes. So does that mean 5,000 go one way and 5,000 go the other, and we hope for the best?
No.
No, as it turns out, the egg is releasing an attractive hormone that makes it more likely that the sperm will go to the correct uterine tube to be able to fertilize that egg. But some could end up taking the wrong path. And of those 10,000 that enter into the uterine tube, only about 200 are going to reach the oocyte. And pretty much all 200 of those are going to be needed because remember, it's not like we see in the cartoons and on the movies. It isn't the first sperm that gets there that wins. As we've talked about, we know that egg has some protective layers on the outer surface. It has that glycogen-rich zona pellucidum. It has those leftover granulosa cells forming the corona radiata. And it takes teamwork to penetrate those defenses so that one single sperm is finally capable of fertilizing the egg. That's why once the sperm levels drop below that 2 million, then basically the number of sperm that reach the egg aren't going to be sufficient to be able to break through the defenses and won't be able to fertilize the egg. Again, this is an illustration, but I do think it does a nice job of showing the point. Here we have those 200 plus sperm fighting to get their way into the egg. And in particular, what I really like about this illustration is it again emphasizes the disparity between the two gametes. Remember, as we mentioned, the sperm basically only brings one thing to the party. It brings 23 chromosomes, and that's it. As we said, it is basically a nucleus with a tail. It is a nuclear missile. As we've also talked about, unlike the sperm during oogenesis, we get that uneven division because the egg that is going to be fertilized isn't only responsible for bringing its 23 chromosomes to the party, but all of the cellular components that allow a cell to be successful and to survive and to live and to divide, the Golgi apparatus, the mitochondria, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, all of those things the egg provides. So for this to be a viable zygote, we have to have all of that cytoplasm. And so that uneven division of the gametes in the female process provides for all of that cytoplasm. And so you see this massive disparity in size between the two gametes. All right, so as I mentioned, for fertilization to take place, and again, by fertilization, I mean successful fertilization that has a chance of implantation and becoming a person, that secondary oocyte after ovulation needs to enter into the uterine tube. Remember, once in the uterine tube, the cilia of those simple columnar cells, that very special epithelial tissue we see in the uterine tube, or what is also known as the oviduct or fallopian tubes, move it about 1/3 of the way down the length to that enlarged region that is the ampulla. Again, it is primarily the cilia and some peristylate contractions that move it. As I said, that oocyte is releasing chemical attractants trying to draw the sperm to it. And in fact, the sperm do swim, as we mentioned, from the vaginal canal through the cervix, through the uterus, into the uterine tube. But remember, we also talked about how the prostaglandins in the semen, as well as a female orgasm, can cause a reverse peristalsis that will help to bring the sperm up towards the oocyte. And remember, this process could occur as quickly as 5 minutes. However, even if the sperm gets there in 5 minutes, it isn't necessarily going to be ready to tackle the process of breaking through the egg's defenses. To be capable of breaking through the egg's defenses, the sperm has to undergo a process called capacitation, where basically it becomes ready to fertilize the egg. And that process can actually take as long as 8 to 10 hours. So basically, right, like any good union, all the workers rush there, and then they have to sit and stand around and wait for several hours before they can actually get to work. Once capacitation occurs, there are basically 2 main processes that this occurs. It is a very vigorous spinning of the tail, which is going to help it work its way through the the corona radiata that is on the outer surface. But remember, we also have that candy-coated shell. And we need to be able to digest those glycogens to be able to work their way through. And to do that, remember the sperm on the tip of the head has that acrosomial cap that contains a large amount of digestive enzymes. However, we don't want to just spill those digestive enzymes any old place. So that acrosomial cap is very sturdy, very strong. However, during the capacitation process, that membrane that holds those digestive enzymes becomes fragile, so that when it actually comes in contact with the zona pellucidum, it will actually rupture, releasing those digestive enzymes and help to eat away at that glycogen-rich shell. Here we have a pretty picture that kind of shows the state of things. Notice in this case, fertilization has just occurred. The sperm has actually penetrated the surface of the plasma membrane of our secondary oocyte. Notice we can tell it's a secondary oocyte because our first polar body's in there. So we know it has already undergone the completion of meiosis 1. And remember, it was frozen in metaphase 2. However, now that the egg has been fertilized, meiosis can now complete, separating into an additional polar body. And then we will have the remaining ovum with half of the chromosomes and now the chromosomes of the male, and that will eventually become our zygote. However, notice the other thing that this illustration shows are those defenses we've talked about. We have the corona radiata, which are the granulosa cells that are surrounding the outer surface of the egg. And we also have that glycogen-rich zona pellucidum, that thick candy-coated shell protecting the egg and keeping it inside. Now, on the surface of the zona pellucida is a very special protein receptor known as the ZP3 receptor. I'm not sure what happened to ZP1 and ZP2, but the important one for us is the ZP3. Remember, I mentioned part of the capacitation process is that acrosomial cap becoming fragile. Well, when the head of the sperm comes in contact with that ZP3 receptor, that is what triggers the release of those digestive enzymes. So all of those, and I'll change colors here. All of those digestive enzymes are released right here in that location. And remember, we talked about how the tail will spin very vigorously, basically grinding the head of the sperm and those digestive enzymes into the zona pellucidum, helping to chip away at that surface. Now, do you think the sperm is going to be able to maintain that very vigorous sperm tail twirling for a long period of time?
No.
No, it's going to dig away a bit at that zona pellucidum, but eventually it is going to run out of energy, run out of fuel, and basically just drift away to die. And then the next one will come in and it'll start drilling away and then go away and die. And the next one and the next one and the next one. Dozens, if not hundreds of sperm are going to be needed to chew away at that membrane until finally one late coming America sperm, right, slept in, went into Starbucks first, got his coffee while all the other ones were doing his work. And after they've all done the work and killed themselves, he gets to come, slide on through, and fertilize the egg. Like I said, nothing more American than the fertilization process. This process of rupturing the acrosomial cap and releasing those digestive enzymes and that digging of the sperm head is what we call the acrosomial reaction. Let's put this all together again. So again, our sperm head binds to that ZP3 receptor, causing the rupturing of the acrosomial cap, releasing those digestive enzymes. And again, it is going to vigorously spin its tail, chewing away at that zona pellucidum, trying to form openings. And obviously, with hundreds of sperm, this is going to be happening in hundreds of different locations, or at least dozens of different locations around the surface of the egg. Until eventually, enough of them working together penetrate the defenses. So again, as we talked about, it's not like in the cartoons, it's not like in the movies. It's not the first one there that wins. It's not the last one either, but it's practically the last one, right? After a lot of work has been done, working together as a team, one of them is finally able to come in. And when it comes in, it makes contact with the plasma membrane of the egg. And that's the second, right, fallacy. We think of it as the sperm fertilizing the egg. That's not actually what happens. The head of the sperm actually comes in contact with the plasma membrane of the secondary oocyte and basically sticks there. And then it is the egg that basically brings the head of the sperm in, wrapping its plasma membrane around it, basically that process that we know of endocytosis, where something attaches to the plasma membrane wraps around and brings it in. And it is actually the egg that actually draws the head of the sperm into the secondary oocyte. Now, we only want one sperm to be able to get in. And this is one of the key ways that we're going to make that happen. If penetrating the sperm, if penetrating the egg was a process of the sperm, then multiple sperm might be able to get in at the same time. But instead, as I said, they come in contact with the plasma membrane, and the egg is the one that draws it in. That drawing in of the single sperm causes a reaction that we call the fast block. This fast block happens almost immediately upon contact of the sperm to the plasma membrane. The first thing that happens is the plasma membrane depolarizes. This depolarization makes it so that basically a second sperm is not able to attach to the plasma membrane. And this depolarization also causes the release of calcium from the endoplasmic reticulum. Remember, we talked about in muscle tissue how the smooth ER, or what we at the time called the sarcoplasmic reticulum, stored a large amount of calcium. Calcium, of course, makes cells do wonky things. And in our muscle cell, that calcium causes the movement of the regulatory proteins, allowing the muscle to contract. Well, in a similar fashion, calcium is stored in the smooth endoplasmic reticulum of the secondary oocyte. So when depolarized, voltage-gated sodium channels open. Calcium channels open. And when those voltage-gated calcium channels open, calcium rushes into the cytosol. And of course, as we know, calcium makes cells do wonky things. In this case, that wonky thing is the slow block, or what we call the cortical reaction. With this cortical reaction, basically three things occur. The first thing that occurs is the releasing of the zonal inhibiting proteins. These are vesicles containing special proteins that do two things. They inactivate the ZP3 receptor. This means that no more sperm will break their acrosomial cap. And none of them will start that vigorous swimming. So it stops the acrosomial reaction. So let's actually say it that way. So no more sperm are going to try to fertilize the egg. And that protective zona pellucida is important. We actually want to maintain it to protect this cell. But remember, all the sperm have been beating away at it, weakening it. So the other thing that that zonal inhibiting protein is going to do is cause the hardening and the thickening of the zona pellucida again. Because we want to, oh, I did this really badly. There we go. We want to maintain that egg shell until it actually gets to the uterus. It won't be until it gets to the uterus that it will hatch. from the zona pellucidum and thereby be released and capable of implantation. That takes care of the extra sperm, making sure no more sperm fertilize the egg, because if that happens, we would have a condition we called polyspermy, multiple sperm in it, which would give us the wrong number of chromosomes and would cause problems. Our goal is to have syngamy, just one sperm fertilizing the egg. So 23 egg chromosomes plus 23 sperm chromosomes equals our diploid zygote. Oops. Eventually. So again, we've made sure that we're only going to get the one sperm in there, but remember, we still have to complete meiosis 2. And so then after meiosis 2, we are going to have our second polar body. And we are going to now have an fertilized ovum. Notice it is not a zygote yet. It is just simply a fertilized ovum. All right, so here we've talked about how we fertilize the egg. Again, maintaining syngamy, making sure that just one sperm and only one sperm gets in. And here we've done it with all the pretty words. Here, you see this great illustration from your textbook where it does a really, really nice job of showing this process. Notice, again, we have the sperm with their acrosomial caps. This way. Working their way through the corona radiata and coming in contact with our ZP3 receptors that are on the surface of the zona pellucidum. That causes the acrosomial reaction, the release of the digestive enzymes, and the vigorous twirling of the tail, grinding the head into the zona pellucida, using those digestive enzymes to break it apart. Doesn't show it here, but again, many sperm are needed to come in, work at that, and come away until finally one sperm is able to make it all the way in. And when it makes it all the way in, it attaches to a binding protein on the surface of the plasma membrane of our secondary oocyte. Notice the head does not actually penetrate the secondary oocyte. It attaches to the surface. Then the egg is active in endocytosing the head of the sperm. As that does it, we get our fast and our slow block. First, we get the big depolarization. That big depolarization leads to the release of calcium. And that release of calcium leads to the slow block, or remember what we said was the cortical reaction. This is when we're releasing that zonal inhibiting protein to basically inactivate the ZP3 receptors and to rethicken the wall of the zona pellucidum to continue to provide that protection for the fertilized egg. And then, like we said, it also has to complete meiosis. And then the process is complete. We now have a fertilized ovum. We finally have a mature female gamete to match our mature male gamete. Questions on that? All right, so how do we get from a fertilized ovum to a zygote? Well, at first, as we can see here, we have that single cell, that ovum, that has completed meiosis 2, and it has its own haploid nucleus. We also, from the head of the sperm, have also brought in the male haploid nucleus. These two nuclei, because they contain half of the number of chromosomes, we call, again, haploid nuclei, as they've been here by themselves, or now that they're both contained but separate inside of our oocyte or ovum at this point, we can now also refer to them as pronuclei. At which point, this cell starts mitosis. And what is the first stage of mitosis?
Phase.
Prophase. Excellent. And what are the things that occur during prophase?
The chromatin condenses into chromosomes.
Okay, we form chromosomes. What else?
The nuclear envelope gets broken apart. And then the mitotic spindle forms.
True, absolutely. So absolutely, we form our chromosomes, our chromatin folds up into chromosomes, our mitotic spindles form, and they're moving to the poles. But the big deal for us right here is that third step. The nuclear envelopes start to break down. As the nuclear envelopes start to break down, we no longer have two pronuclei. We now just have this cluster of chromosomes that are floating around together. And once those nuclear envelopes break down and all the chromosomes come together, that is when we have a zygote. So now that all the chromosomes have come together, now we can consider this cell a zygote. And again, I think I actually have some pretty pictures that show this. So it's going to form that fertilized ovum, that diploid cell forming that zygote. Here we go. So notice here we see fertilization taking place. Again, the sperm has made contact with the outer surface. It has been drawn in, and its little nucleus is now inside of the egg. Our secondary oocyte is completing meiosis 2. And notice also our polar body is completing meiosis 2. So at the end of that process, notice we have 3 polar bodies and our mature ovum. These then start the process of undergoing prophase. Notice our 2 spindles are forming and migrating to the poles. Our chromatin is condensing down into chromosomes. And notice once the nuclear envelopes break down, all the chromosomes have come together. And basically all the chromosomes, once they're together in that one single cell, we now officially have a zygote. All right. Questions on that? No, notice it forms this zygote as it is undergoing mitosis. And when it completes mitosis, what are we going to have? That one zygote is going to become what?
2.
2 zygotes? Is that what we would call that at that point? One zygote divides to become 2 cells, and those 2 cells are 2 zygotes?
Isn't it like a blastocyst?
Close. It is a absolutely. It is, as it divides, it becomes what we call, oops, I thought I had the term here. There it is, blastomeres. So the individual cells, so let's cheat and come back here for a second because I want to get back to this other point. When the one zygote divides to become 2 cells, all of these individual cells are individually called blastomeres at this point. So now we have 2 blastomeres. And of course, those 2 blastomeres divide to become 4 blastomeres. Those 4 blastomeres divide to become 8 blastomeres, and so on and so forth. And one of the important things to remember, well, actually, I lied. There's 2 important things to remember about this division. And I'm going to cheat and steal some of the board up here so I can write this. The first is these are very rapid divisions. Why is that important? The reason is there's not a lot of time. And let's be scientific about this. For G1 stage of interface. Someone remind me again what happens during the G1 stage of interface? It's okay, I don't remember it. No one, really?
Does the cell get bigger and store nutrients?
Yeah, remember, basically G1 is the stage where the cell's getting ready to divide, primarily by getting bigger and making a lot more endoplasmic reticulum and making a lot more Golgi body and making it a lot more mitochondria and all those things, so that when those 2 cells divide, the 2 divided cells are pretty much the same size, if a little bit smaller than the original cell. So the cell gets huge in anticipation of becoming 2 cells. Well, in this case, the cells don't have time to make a lot of cytoplasm, so their size doesn't really change. So notice, like this ball that I have here that I've sliced up like a pizza, when one cell becomes 2, those 2 cells are basically half the size of the original. When those 2 cells become 4, they're half the size of the original. So notice, as a result of this, we get more cells, but each time the cells get smaller. So the overall size stays the same. And this is important because of the second point I want to emphasize. Remember, this is all still occurring inside the zona pellucidum. So there's no room for the cell to get bigger because it's still inside of this candy-coated shell. And again, my drawing is horrible, but we see this nicely on the next page. However, there is one point I want to emphasize. There are basically 2 different types of twins we can have: monozygotic or dizygotic. Dizygotic are what we typically call fraternal twins. What are monozygotic twins also known as?
Identical twins.
And what's the difference between the 2 types of twins?
The identical twins come from the same zygote, and then fraternals are 2 different zygotes.
Absolutely. With identical or monozygotic twins, we have one egg, one sperm, one zygote. However, with identical twins, what happens? Yeah, let's move these up here. As we are forming those blastomeres, the ball of cells splits into two balls of cells. And each of those balls of cell forms its own person. So you have two people with identical genetic information. Right? Whereas with dizygotic twins, oh, and I should say this identical twins is pretty much a random act. And technically can happen to pretty much anyone. Dizygotic twins are much more commonly run in families, especially on the female side. And the reason for this is that some women are more likely to ovulate two eggs. And if you ovulate those two eggs, each egg is of course different, but each egg is individually fertilized. And obviously they're going to be fertilized with two different sperm. And so basically you have two completely different people growing inside of you at the same time. Right? This is how you can get a Brenda and a Brandon, because one egg could be fertilized with a X chromosome sperm and the other could be fertilized with a Y chromosome sperm, giving you brothers and sisters. In fact, there's been at least one in multiple recorded cases of individuals who have actually given birth to half fraternal twins, whereas both eggs were released by mom, but each egg was fertilized by a different sperm donor. So one male provided the sperm for one egg and a different male provided the sperm for a different egg. And so basically these two babies form in the belly together, are birthed at the same time, and are actually half siblings, sharing the mom with different dads. Again, I'm not encouraging this, but it has occurred. All right. Do we understand those distinctions? Excellent. And lastly, let's go back up here. As I mentioned, we form those blastomeres and they split into two independent balls of cells, forming two independent people. But sometimes this split occurs later in the developmental process and the two balls don't completely separate from each other. What happens in that instance?
Conjoined twins.
Conjoined twins, absolutely, where basically the bodies don't form completely independently. They can share internal organs, like share a liver or share an intestinal tract, right, where they don't completely separate from each other, forming those conjoined twins. Excellent. All right. As I mentioned, and here we see that division. Again, it's a rapid mitotic division, giving us identical cells, but because it's so rapid, the cells don't have a chance to grow. Notice that zygote divides into our first two blastomeres, usually within the first day, about 30 hours after fertilization. By day 2, our second cleavage has occurred. This is what we call the division of these cells, the rapid division of these cells, forming 4 blastomeres. By day 3, we usually have a structure that it is a solid ball of blastomeres. This solid ball of blastomeres is known as a morolla, named after the mulberry, because that's kind of what it looks a little bit like. And then around day 4, you have a solid ball of well over 100 cells. Now remember, during this time, from day 1 through day 4 of this development, this cleavage process that is taking place, at the same time, the developing cluster of cells, again, we'll call it an embryo at this point, is migrating down the uterine tube. Right? Those cilia and peristaltic contractions are moving it towards the uterus. And finally, day 5 occurs. When day 5 occurs, basically 2 very important processes have taken place. By day 5, we have finally made it to the uterus. Again, once in the uterus, as we talked about, the uterus has been preparing, thickening the functional wall, vascularizing the functional wall, and producing and releasing that uterine milk to help to maintain this bundle of cells. But something else magical has happened as well. Remember, up to this point, it has been a solid mass of cells, all completely uniform. But by day 5, our bundles of cells have formed a special structure known as the blastocyst. Notice, as we look at the blastocyst from the outer structure, it looks like a solid ball of cells. But in fact, if we were to cut it in half and look inside, we see that that is not the case. What we're seeing from the outer surface is a solid layer of cells that are forming the outer structure of the blastocyst. But notice we're finally seeing some differentiation on the inside. In one portion of the blastocyst, we are getting a dense clustering of cells. All these cells tightly packing together, forming what is known as the inner cell mass. And we have a fluid-filled space. This ball is actually mostly hollow. And this fluid-filled space is known as the blastocyst cavity. Lastly, I mentioned them, but I didn't give them a label. The cells that form the outer shell of the blastocyst is the trophoblast. So actually, let's change the color of that to red so it's consistent. So we have an outer layer of cells. All these cells that we're seeing here on the outer surface is the trophoblast cells. Then we have our inner cell mass and a big hollow space known as the blastocyst cavity. Now, remember, as we mentioned, once we get to the uterus, that uterine milk that is being produced is going to help to maintain this blastocyst. And it is at this stage, as a blastocyst in the uterus, that it is finally going to hatch from the zona pellucidum. Oops. And that's what's going to happen. Once we get into here, it hatches and then it implants. I found this nice video on the almighty YouTube. That does a nice job of showing this process. So again, notice we can see the zona pellucidum on its outer surface. And basically we have a single cell inside of there. Oops, put this up and out of my way. And there's our first division. Our zygote has divided into 2 blastomeres. Those 2 have divided into 4. And notice they're also starting the 3rd cleavage formation as well. And again, notice with each division, the cells get smaller and it is still contained within the zona pellucidum. Notice as more and more cells divide, it becomes that morella, that solid ball of cells. From 16 to 32 to 64, and on till we have a full cluster. And I went a teeny bit too far. A solid mass of cells. However, notice you're going to start to see some movement of the cells. Notice cells are now moving out to the outer surface and condensing on the inside. So notice here we have our trophoblast on the outer surface. We have our inner cell mass here along the inside wall and a big fluid-filled cavity. However, notice I also stopped it a second too late. Notice now, again, all of this has been occurring inside that zona pellucidum. But notice it is finally punctured through the zona pellucidum and is actually in the process of hatching. And as you notice, when it hatches and comes out, after being condensed in the zona pellucidum so long, it is going to expand in size and overall become a larger structure. So here we see it forcing its way, hatching out of that zona pellucidum. Oh, and apparently that's all we get to see. We don't get to see the end of the process. But you saw it at least halfway out of the zona pellucidum, and then they didn't show us the rest. All righty. Questions on that?
Professor, at
Yes.
this point, those cells are pluripotent or
Omnipotent. At this point, these cells are omnipotent. Even, well, okay, let's say it this way. Up to the morella stage, every single one of these cells is completely omnipotent. Here, when we start to get to the level of the blastocyst, this is where we're going to see a little bit of a change. The inner cell mass is still basically omnipotent stem cells. However, the trophoblast is starting to change. These cells are going to have different destinations. So let's clear my drawing here for a second. Let's say, for instance, I were to take this cell and switch them. That person's not going to suddenly have a hand where their foot's going to be and their foot where a hand was going to be. That person would be completely normal because those are omnipotent cells that, again, can become anything and are going to become anything. However, if I were to change, say, this cell with this cell, depending on how far developed that blastocyst is, that could cause developmental problems. Because what the trophoblast becomes and what the inner cell blast becomes is going to start to be different. So it's at this point we start to see some differentiation, but still most of the cells are still omnipotent stem cells until basically invagination occurs, which is a process we're not going to talk too much about, but we'll briefly show it. But yeah, definitely up to this point, all of these cells are omnipotent and can become anything. That's how, when these split, you get two identical people. You don't get two half peoples. You get cells that are able to divide to become everything necessary to make a person.
Oh, that makes sense. Because previously I asked about that meiosis lab we did, and it was like exactly the video you just showed us, and they took only, for example, if there was 8 eggs in the blastomere, they took just one of them to do that IVF thing. That's why I was confused if we can get in different individuals from one blastomere.
Yes, well, yes. So absolutely. So from, or at least from the morella, yes. If you were to take these cells and develop them independently, they could potentially come different, identical people, but two different people. Yes.
Okay, thank you.
Yep. All right, excellent. How are we on time? Actually, you know what? We don't have a ton of material to cover. So, and the next part's got probably the worst vocabulary you're going to be responsible for. So even though it's a teeny bit early, let's go ahead and take our first break here. It is 12:53, so we'll come back at 1:08. And I will start the recording at that time. At that point, like I said, we will talk about the process of implantation, briefly mention the specializations that are going to take place. And then from there, like I said, fast forward to the end of the process. So, all right, let's go ahead and take our first break. I'll see you guys back here in 15 minutes. All righty. Any questions before we dive back in? All righty, we've been working our way through this process. We have fertilized the egg. It is migrated to the uterus. It has undergone cleavage to form our blastocyst. And at the end of what we talked about last time, it now finally hatches from that zona pellucidum and is free floating within the uterus. Now, at this point, it doesn't instantaneously implant into the uterine wall. Remember, the uterus is filled with that uterine milk that we talked about, that massive glandular secretions, and it can be maintained in there for another 2 to even 4 days. But eventually, usually around day 7 or day 8, it will finally attach to the simple columnar epithelial tissue on the outer surface of our functional endometrium. Now, again, notice up here we have our illustration of the uterus. Ideally, implantation will take place in the body or along the fundus of this. If implantation occurs too close or even over the, well, let me rephrase that. If it occurs too close to the cervix, then as the placenta forms, the placenta can actually form over or too close to the opening of the cervical canal. And then when dilation would occur in anticipation of giving birth, that could actually cause the placenta to rupture, causing serious problems for both mom and baby. So typically, if that occurs, if the placenta forms too close to the cervix, then typically that mom will be monitored much more closely during the pregnancy, and the baby instead will be delivered via cesarean. So a traditional childbirth will not be an option in that situation because of the potential risks of bleeding from that. But in most cases, it is going to implant, like I said, into the body or the fundus, into the functional layer. Now, notice the first step in this process is the blastocyst orients, so that the inner cell mass is actually facing the uterine wall. And once it is facing the uterine wall, it'll make contact with it, attached to it, and actually start the process of penetrating deep into the functional layer to be surrounded by the tissue and protected. Now, obviously, it needs a way to drill into that functional layer, and here we see how that occurs. Remember that outer cell layer, the trophoblast, which we'll highlight here in green, undergoes a change where basically two distinct layers form. Here in light green, we'll show the portion that stays intact, continues. Okay, that doesn't differentiate it enough. So let's use, so many colors on this. We'll use red. Stays intact, maintaining the integrity of the cavity that is formed by the trophoblast. And that region is what is known as the cytotrophoblast. So the cytotrophoblast is basically helps to maintain the original shape and structure of our blastocyst. However, the more interesting portion of this, and even though it's yellow, I'll use orange to emphasize it, portions of the trophoblast expand out. And as they expand out, they become almost glandular in nature, where they're producing and releasing digestive enzymes. And these digestive enzymes start to break down the functional wall, basically dissolving the functional wall to form a space that the blastocyst can implant itself into the functional layer. And this specialized region of the trophoblast is called the Incidiotrophoblast. And I'm sure as you sit here staring at that word, you know with 100% certainty you're going to have to spell that on either the lab or the lecture exam at some point. All right. This Incidiotrophoblast actually does a couple of things. As it is, let's see what the next slide looks like, see if that helps. Here we go. Excellent. As the Incidiotrophoblast continues to expand, forming these digestive cells that are digesting the wall of the uterus, notice they are also going to be breaking down all of those big blood vessels we talked about as well. So it continues to dig its way in. And as it is continuing to dig its way in, it is basically going to form two structures that are going to form the foundation of the placenta. Here, let's do this way. I'm going to draw this. So here's the functional layer. And as the Incidiotrophoblast is forming these extensions that are extending down into the functional layer, not only is it forming a cavity that the blastocyst can embed itself within the walls, but as I mentioned, it is also dissolving the functional tissue of the functional layer. And part of that is blood vessels. So, oops, I wanted to change the colors there. As we digest that tissue within here, what ends up happening are these spaces occur. These spaces are connected to all the big blood vessels, and these spaces basically become a big blood reservoir. So these finger-like extensions extend out, becoming these finger-like extensions, these papillae-like structures. They dissolve the functional layer. They form this reservoir of blood, and then blood vessel capillaries are going to form here in these papillary fingers. And it will actually be here between the blood reservoir of the mom's uterine wall and these capillaries that the exchange of materials will take place. This is how baby will get its oxygen and its nutrients. This is how the baby will get rid of some of its wastes. And this is the structure that ultimately will come together to form the placenta. Notice that the blood supplies of mom and baby do not mix, right? Which is a good thing. We don't want mom's antibodies checking out baby's cells and seeing them as not being correct. And by the same token, the things that baby's making, we don't want messing up mom either. But this is how we need to get our oxygen. This is how we need to get our nutrients. This is how the baby needs to get rid of its CO2. So this exchange takes place. And the advantage of having this big blood reservoir is, despite what people think, are pregnant women eating all of the time? Of course not. Are they always eating the most nutritious and well-balanced meals all of the time? No, but the baby needs a constant supply of resources. By having this blood reservoir, it can contain a large amount of nutrients, a large amount of oxygen, a large amount of chemical signals and hormones, so that while there may be dynamic fluctuations in what's going on in mom's systemic capillaries, those systemic capillaries are constantly filling into this pool. This pool is constantly draining, and so it maintains a more consistent environment for the exchange. So regardless of whether mom's sleeping, whether mom's exercising, whether mom's eating, the baby is basically consistently getting the same type of materials that it needs in a fairly regular pattern. And again, I've done a truly horrible job of drawing this, but let's look at the pretty pictures from your textbook that show this. Again, remember, we're able to maintain the uterine wall because the cells of the trophoblast are producing and releasing that human chorionic gonadotropin. And notice we say it maintains the uterus, but as we know, really what it does is it maintains the corpus luteum. And by maintaining the corpus luteum, we keep progestin levels high, and keeping progestin levels high helps us to maintain the uterus. Here's the picture I wanted. Notice that Incidiotrophoblast doesn't just make the hole where the trophoblast is able to embed itself inside. And notice it does truly embed itself inside. Notice the epithelial layer reforms on the outer surface. So it is completely contained within the functional wall of the endometrium. But notice also, as it dissolves away the tissue and dissolves away the blood vessels, we start to get that formation of those blood-filled cavities, what are known as the intervillous spaces. And notice we're starting to get the extensions of these finger-like extensions of the trophoblast, what we call the chorionic villi. And this structure, these finger-like extensions and these fluid-filled spaces, are eventually going to form our placenta. And we'll take a look at that placenta in just a minute. Now, there's one other thing that I want to talk about for implantation. Notice so far, we've only talked about what is going on in the trophoblast. However, as you can clearly see from the illustration, there's a whole lot of stuff going on with that inner cell mass as well. Right, that is eventually going to become, the baby essentially. And again, we'll talk briefly about what's happening there. But as you can see from this illustration and from these illustrations, we are getting some dramatic changes in that inner cell mass to form ultimately the baby and what is going to become the embryonic sac. Now, as you can see nicely from this last illustration, as that inner cell mass grows, it basically, as you can see, forms 3 distinct layers of cells, what are known as the germinal layers. These 3 germinal layers play an important role in the development of the embryo. We have the innermost layer, the endoderm. This is basically when it invaginates and turns itself inside out, is going to become basically the outer surfaces of the baby, forming the epithelium of the gastrointestinal tract and the reproductive tract. So again, remember, those are all systems that are open to the outside world. Our mesoderm, if you remember way back when we talked about muscles, remember mesoderm were the embryonic cells that we said produced our muscle. But they're also the ones that also eventually produce our bone and our other connective tissues as well. And lastly, we have the ectoderm, which becomes basically the outer surface of the body with the epidermis, and also becomes our nervous tissue. Again, this is not an exaggeration. You could spend an entire semester just talking about this process. But as you can see, simply, basically that inner cell mass kind of folds in on itself and extends outward. And as that occurs, it forms basically the amniotic sac and the baby itself. And that process of folding in on itself is what is known as gastrulation. And that's basically how we form all of the organs of the body. And at the same time, we are developing internally the nervous system. We talked a little bit about this in 430, if you remember, when we talked about how you start out with a single layer of cells called the neural plate. That neural plate gets a crease in it called the neural fold. That neural fold allows it to fold up into a neural tube. And those neural tubes form those enlargements that we call the cephalons. And remember, those 5 distinct enlargements of that neural tube become the cerebrum and the midbrain and the pons and cerebellum and all those other structures we learned in 430. And again, whole semester on this stuff, so I'm not even going to attempt to go even a brief introduction of this other than to explain these 2 terms and what they entail. We do want to take a look at that completed placenta. Notice, as we mentioned, we started with those finger-like extensions that now not only have extended out, forming the chorionic villi, but as you notice, these chorionic villi contain capillaries from the baby. And that is where the fetal, because again, once we have a placenta, it is a fetus, that fetal blood is coming for exchange to take place. And notice we also have these big, huge blood-filled spaces. And again, these big, huge blood-filled spaces, as I mentioned, are, as you can see, they're fed into by the arteries, fed out of by the veins, and that is that intervillous space where the reservoir of oxygen, nutrients, hormones, other chemical signals take place for that consistent exchange between mom and baby. And again, the blood cells don't meet. They have and epithelial tissue in between them, but the blood supplies do not meet. All right, so again, we have the big cavities in the functional layer and those intervillous spaces. We have the chorionic villi that were formed by the trophoblast of the baby. And again, it allows the exchange to take place, but doesn't allow the two blood supplies to mix. And let's think about what's moving where. What kind of things is mom giving to the baby? My microphone has been on this whole time, right? I have been speaking. You can't hear.
Yeah.
And my voice went up at the end, kind of inferring that I was asking a question.
Oxygen and nutrients.
Okay, excellent. Anything else? I'm asking the question, so of course the answer is yes. So what else might mom provide for baby via the placenta?
Immunity.
Yeah, in the form of what?
Antibodies.
Which
Antigens.
ones?
IgA.
Close. IgA was the one from the glandular secretions. So which was the one that can cross the placenta? Well, think of it this way. If mom gets sick, gets a common cold, that virus invades her, is there a chance that the virus might invade the baby as well? Yeah, so which immunoglobulin would help us in that situation? Ooh, it's a good thing this isn't a cumulative class where I need to know my 5 classes of immunoglobulins and what they do.
Is it either M or G?
Yeah, primarily G, although probably primarily M and also G as well. Absolutely.
Because M's are the ones that like are immediately like the first ones, right? And then G's
Yep.
are the ones that are kind of like in the background.
Yeah, G's are the ones that are responsible for our primary and secondary hormonal responses. M's, like you mentioned, are the first responders and the ones that can cross the placenta. But again, if mom is invaded, it could also be G as well. And again, this G that can get over is why the whole Rh positive is an issue. Remember, as we talked about, if mom is Rh negative and the baby is Rh positive and mom has built up Rh positive IgGs, those can cross the placental barrier and attack baby's blood. And as we've talked about, that is a bad thing. So that's why we talked about that Rogam and other medications like that that can impact and stop that from occurring. Excellent. What kind of things are going to go from baby to mom?
Wastes.
Okay. All wastes?
Mostly urine.
Well, so let's think about that. I'm so glad you said that. That's awesome. All right, so let's think about this. How do we get rid of wastes in our body? Your body, how do you get rid of your wastes?
Lungs, kidneys, skin,
Okay.
sweat. Oh yeah, that's
Yeah, skin.
skin.
Well, and you could argue digestive, although that's not really getting rid of waste. That's getting rid of, I mean, defecation is getting rid of stuff that we don't want, right? So it isn't so much waste, but things we can't use. Clearly, the baby's not using the digestive system because it's not ingesting any food. So that's not an issue that way, right? Lungs. Do the babies breathe when they're in utero?
No.
No. So absolutely, we got to get rid of CO2 out of the placenta, out of mom's blood that way, right? And there are other wastes that can be taken out of that way as well. But let's think about the kidneys. The kidneys are one of the main structures that are responsible for removing wastes from the blood. And are the kidneys active in utero?
Yes.
Yes.
Yeah, absolutely. Yes, they are. So then where does the pee-pee go?
The amniotic fluid.
Exactly. Amniotic fluid is basically baby urine. For those of you that have been pregnant, one of the things that they closely monitor is the volume of amniotic fluid. If the volume of amniotic fluid is low, then they take a much closer look at the kidneys and the urinary tract of the baby because that can be a concern. So yeah, absolutely. Definitely you are right. There are some wastes that need to be gotten rid of through the blood, but one of the primary ones is CO2 because a lot of the metabolic wastes are actually released via the urine to form the amniotic fluid. So amniotic fluid is basically baby pee. All right, think of that next time you see one of those movies where someone's water breaks and it splashes all over the floor. That's baby pee all over the floor. Doesn't make it quite so cute. All right, excellent. Questions on that? And like I said, the advantage of having that intervillous space, that blood reservoir for moms is, again, it can provide consistent nutrients and more consistent hormone levels, regardless of what activities mom are participating in moment to moment. And like I said, this system also forms that important barrier to keep the blood and therefore most of the cells separate from each other so that we don't have those types of immune reactions that could cause problems. Obviously, when the baby is born, everybody's all super excited about the baby, but we can't forget that the placenta has to detach and release. And in fact, it is very important that the doctor check to make sure that the placenta has detached and released completely. Aside from the fact that it is highly vascularized, and so if it tears during the birthing process, that can lead to an extreme danger of blood loss for mom. But even if there isn't a rupturing of blood vessels, if some of the placenta stays behind, remember it is a very glandular structure producing hormones. And those placental hormones can interfere. Those placental hormones can interfere with the baby's ability to, with the mom's ability to be able to, for instance, produce milk and have other issues that way as well. So it is important to make sure that it is released so mom can recover, mom can produce milk, and then obviously mom doesn't have the blood loss.
Professor?
Yes.
Is placenta really rich of nutrients that I know some, like not so many, but some moms make their placenta into capsules and eat them after birth? So
the capsules is definitely a new thing. There are definitely some cultures and some religions where the placenta is ingested after birth. Basque is a perfect example of a culture where that is a very common ceremony. Others, again, for cultural or social reasons, will take the placenta and bury it with a tree. And so that the tree and the baby both grow at the same time and at the same rate, and there's that bond between them that way. Yes, there are one of the things that you see in the animal world is in the animal world, most mammals, the mom will ingest the placenta. And it is believed that is done for two reasons. One, as you mentioned, it is very nutrient rich. That it has that way. But remember also, there's a massive amount of hormones in there as well. And so they believe that animals ingest it because those hormones in there can help to stave off postpartum depression. So those, and because obviously with the loss of the placenta, again, the female is going to have major fluctuations in her hormone levels, and it is not uncommon for some women to have difficulties because of those changes in hormone levels. And so it is believed that that occurs that way. Converting it into capsules, again, I guess it depends on how you do that. If you're burning it and grinding it up, or drying it and burning it and grinding it up, that wouldn't work. But drying it out and grinding it up, I think the advantage is that it makes it more palatable. I don't know if that necessarily retains the nutritional components of it, but I would say that for most people, again, they're so busy worrying about the baby that they spend little to no account on the placenta and the umbilical cord after it's cut, and in most cases it's just, you know, left as medical waste. That is a great question, Maximilian, sorry. It really depends. Some women, it can take, you know, a couple years before hormone levels are back to normal again, especially because of the fact that breastfeeding also affects the hormone levels in the woman, and those can affect things as well. So some are able to bounce back. And again, it's really, in many ways, it's not dissimilar to the process of the normal ovarian cycle. Some women are able to tolerate the wild fluctuations that we saw in hormone levels from cycle to cycle, whereas some women have much more difficulty getting, you know, PMS and premenstrual syndrome, where they get, you know, horrible headaches, or, you know, they're very fatigued or very irritable, or things along those lines as well. I mean, those same changes are taking place in every woman, but for whatever reason, some women are more sensitive to those changes than others. So it would be the same thing for reestablishing the normal hormone levels after. Some women are going to be more sensitive to it, and others are not going to be as sensitive to it. So there is no set time. The right amount of time is however much time it takes for her to get back to feeling normal again. That is the right amount of time. That is true, absolutely. Again, I think it is very clear that if it was reliant on males to give birth, that this species would have died off long, long ago, because we're big wusses. Again, the placenta is connected to the umbilical cord. There are actually 2 arteries and 1 vein. And again, normally when we think of these arteries and veins, we think of them in, as we know, in the relation to the direction of movement of the heart. So in this case, the arteries and veins in the umbilical cord are baby blood vessels. So what that means is that the arteries move away from the baby heart, and veins move towards the baby heart. So which of those, the 2 arteries or the 1 vein, is going to be oxygen rich in this case? For
The
baby.
vein?
Yeah, exactly. It is the one that is picking up the oxygen from mom and bringing it back. And just out of curiosity, where does the vein go?
Liver.
Exactly, right? It goes to the liver, right? If you think about it, that makes sense. It is picking up all of those nutrients from mom. And as we know, depending if mom's been eating nothing but pickles and ice cream, it may be nutrient rich, but not necessarily nutrient appropriate. So it has to go to the liver first to be processed. Or maybe when we talked about the digestive system, you remember how we talked about the round ligament, and how that round ligament was basically the vestigial structure left over from this umbilical vein traveling its way back. Hold on, looks like there was a question or comment here. Great question. So there are several things that can cause it. If it is during the birthing process, again, the contractions that are taking place can actually cause the placenta to separate from the uterine wall. So if that were to occur, that could cause a tearing in that. Or as we mentioned, if it is too close to the cervix, as the cervix is dilating and effacing, that can cause a tearing of the placenta as well. And notice, as we just mentioned, the placenta is responsible for bringing the oxygen to the baby, right? It gives the oxygen to the vein, and the vein carries it back to the baby's liver, and then obviously to the heart, and then everywhere else from there. So if that placenta is damaged, then there's not enough oxygen being transported in this vein back to the baby. And then, of course, also, as we mentioned, the artery is going to be oxygen poor, going to the placenta, and also will be CO2 rich. And I think I've got a pretty picture that shows this. Yep. Notice here we see the two arteries. The two arteries actually come off the internal iliac, come up and travel to the placenta. And then that one large oxygen-rich vein, as we mentioned, goes straight to the liver. And when the umbilical cord is cut on the outside, this all forms the belly button. And on the inside, this shrivels up and becomes the falciform ligament. I'm not the falciform, the ligamentum rotundum, the round ligament. So there we see that. So that would be my guess as to what happened, Dan. Maximilian, we are glad that you are okay. All righty. Questions on this or more comments? All right. Excellent. And we did that already. Round ligament. Perfect. All right. So we now know how to implant the egg. We know how to make the placenta, what the placenta does. And like I said, we're fast forwarding through all the rest of this until it is time for parturition, basically labor and delivery. Now, as you can see here, we can see the hormones that are maintained and maintaining the pregnancy. Notice again, we have the things that we expect. Estrogen and progestin levels are extending, going up, primarily progesterone levels. Those are at first, prior to fertilization, being maintained by the corpus luteum and luteinizing hormone. But remember, our trophoblast is making that human chorionic gonadotropin that is going to maintain the corpus luteum until the placenta forms, at which point human chorionic gonadotropin levels start to drop. As the placenta grows and gets bigger, the hormones grow and get bigger as well. However, notice something interesting happens around week 24. Up to this point in time, progesterone has been the higher level of hormones, but now it's still going up. But notice estrogen is now going up even more. And then notice right at the time of birth, estrogen continues to spike and progestin levels start to drop. So again, we're seeing these dramatic changes that are taking place in the hormones of the woman during pregnancy. Again, our trophoblast is producing human chorionic gonadotropin, the ovum is. Our corpus luteum produces the progestin and the estrogens until the placenta, again, starting around week 9 till it's fully formed by the end of the 3rd month, is the primary source of our estrogen and progesterones. However, as we also know, the placenta produces a lot of other hormones as well, like relaxin. What did we say relaxin does?
Relaxes the pelvic muscles.
Well, it relaxes the ligaments. Remember, we've seen like that broad ligament and the round ligament and the uterine ligament. All of those ligaments relax, and the other ligaments that you're right, like the fascias that are holding the muscles in place, all relax to allow for the accommodation of that growing baby inside. You also remember the pubic symphysis, which is that fibrocartilage joint of the pelvis. Relaxin also loosens that as well to make it easier for the baby to pass through the true pelvis. And as we also talked about, because the pelvis is our center of balance and equilibrium, it also tends to give, especially late-term pregnant women, a very distinctive gait in the way that they walk, right? And I didn't say waddle. But there are a couple other hormones that are being produced as well. One of those is human chorionic somatomammotropin, which is a mouthful, or also known as human placental lactogen. You may use either of those two terms to describe it. But with a name like mammotropin, with a name like lactogen, what do you think this hormone does?
Matures the lactiferous glands.
Yeah. Every woman and every man have lactiferous glands in their mammary regions. However, if you are a male, or if you're a female who has never been pregnant, those mammary glands, those lactiferous glands, are completely immature. They are incapable of producing milk. However, this human chorionic somatomammotropin causes the maturation of those mammary glands. This is often associated with a swelling of the breast that occurs during pregnancy, as well as the breast becoming more tender, especially on the first pregnancy, because these glands are growing and expanding. Notice they're maturing. They're not producing milk yet, but they are becoming capable of producing milk, right? We know the hormone that is responsible for making the milk. What hormone again is responsible for making milk?
Prolactin.
Excellent. And there's even a hormone that helps the woman to let down that milk so it's easier to express. What hormone was that again?
Oxytocin.
Yeah. So those are milk production and milk release. That's not what we're talking about. We're just talking about getting the mammary glands ready for production. And so that starts with the formation of the placenta, the formation of this human chorionic somatotropin. And like I said, it usually reaches its peak around week 32, which makes sense because the baby is pretty much viable from week 36 on. Lastly, another one of those hormones that has a lot of major effects is corticotropin releasing hormones. What were the corticotropins again?
The hormones released from the adrenal cortex.
Yeah, and if you remember, the hormones released from the adrenal cortex played a really important role in mobilizing energy, getting that fuel out, getting that fuel ready, typically to deal with a stress situation. But in this case, it's helping mom to be able to deal with the massive amount of metabolism that is going on inside of her body, right? They become little baby ovens. One of my favorite stories my wife loves to tell is when she could really knew for the first time that she was pregnant is we had gone out because, you know, we were celebrating and telling everybody about it. And we had gone to a cheesecake factory with some of the grandparents. And anybody who's ever been inside a cheesecake factory knows they keep that place at about -3 degrees Celsius on the inside of a cheesecake factory. Those places are freezing cold. And so all of us were in there shivering and needing blankets and all of that. And my wife was just happy as a clam because her metabolism was so high and her energy was so high. She was like this little baby oven. And again, this helps to release those resources, metabolizing the fat, increasing metabolism of the body, and helping her to release those resources to get it ready. Well, not to get it ready, to help in all of that development that is going on inside. Corticotropin releasing hormone also plays a role in the induction of labor as well. So let's talk about that. Childbirth, parturition, or labor are the series of events that expels the infant from the uterus. Again, notice as we saw on that previous chart, there is a dramatic increase in estrogen levels that is coupled with that drop in progestins. So progestin levels start to tail off, but estrogen continues to rise. As I mentioned, that corticotropin releasing hormone level rises and reaches a peak. And when that occurs, that triggers uterine contractions. And as we know, these uterine contractions produce a positive feedback process. We should know this because we talked about this way back in 430, but let's go ahead and do it again. We have a reflex arc. With any reflex arc, we basically have those 4 main components. We have a disturbance. We have a receptor that perceives the disturbance. We have a command center that processes that information, makes sense of that information, compares it to what we know, and makes a decision, and then sends that information to an effector. Oops. So in this case, what is the disturbance?
The infant's head pushing against the cervix.
Yeah. The infant's head pushes against the cervix. That stretches the cervix. When the cervix stretches, there are, whoa, way too big, and a one-way arrow. When it stretches, there are stretch receptors that perceive that information and send action potentials to the command center. Our command center in this case is the hypothalamus. And the hypothalamus does what?
Does it release gonadotropin releasing hormone?
Close. What ultimately do we want to be? Ultimately do we want release? We want to release what? What's the hormone that's going to keep this positive feedback system going?
Oxytocin.
Oxytocin is released from which part of the pituitary gland?
Posterior.
Yeah. So how does the hypothalamus control the posterior pituitary?
Neurally.
Yep. So our hypothalamus sends action potentials to the posterior pituitary, and our posterior pituitary releases oxytocin. What does that oxytocin do?
Contractions of the uterus.
Causes a contraction of the uterus. And notice when you cause a contraction of the uterus, that is going to lead to an increase of the force that is pushing the baby's head against the cervix. That is going to cause baby's head to stretch the cervix even more, send even more action potentials to the hypothalamus. The hypothalamus is going to cause the anterior pituitary to release even more oxytocin, and the uterus is going to contract even stronger, pushing baby's head against the cervix even more. And again, notice, like we've talked about, with a positive feedback process, each time the disturbance is increased. Now, remember, our goal is still the same. What is our goal of any feedback process?
Maintain homeostasis.
Yeah, we want to bring the body back into balance. Well, how does this process bring the body back into balance?
Getting the baby out of there.
Exactly. Once the baby is expulsed from the uterus, then it's no longer pushing against the head of the cervix, and the body is able to reestablish homeostasis. Life is no longer in homeostasis, but at least the body is back in homeostasis. Again, if you took me for 430, we talked about this on the second day of 430. And here we are pulling back to this process that we talked about then. I think that's called the circle of life. But don't worry, I'm not going to sing. Excellent. Now, remember, these changes, the increase in the estrogen, the corticotropin releasing hormone spiking, all of those things, just the baby getting big and pushing and being in the right position can cause that. But remember also, we talked about prostaglandins. Prostaglandins can be released by the ejaculate in the male, which is why we mentioned you have to be careful about having sex during late terms of pregnancy, because if we start the uterus contracting, it can start this positive feedback process. By the same token, even if ejaculation doesn't occur into the female, right, you want to be careful about bringing the female to orgasm, because again, it can cause those uterine contractions. And the placenta itself is going to release prostaglandins to help to start that process by triggering those uterine contractions. All the goal is to get that uterus contraction starting so we can start that positive feedback process and ultimately expulse the baby from the uterus. All right. Questions on that? So that is the goal, but parturition basically occurs in a 3-step process because nothing can ever be just that simple. It starts with dilation. I still didn't grab a ruler after the last time. Shucks. All right, well, you're just gonna have to look at what 10 centimeters is to see how small and tiny that actually is. As we mentioned, upon the onset of labor, you get a dilation of the cervix. Remember, we talked about how there's a mucus plug in the cervix that helps to provide protection prior to and during pregnancy. That we talked about how it thinned during ovulation to make it easier for the sperm to get through. As it dilates, that mucus plug is going to be released. And not only does it enlarge, but the walls become more pliable. They efface. So they become a softer and more pliable. So again, the doctor or the nurse checks manually the diameter of the cervix and how soft it has become to reach that stage for parturition to occur. At this point, uterine contractions typically begin, mildly but slowly building. And of our 3 stages, this is the one that is most variable. In a female who has been pregnant before, dilation can occur in as little as an hour to an hour and a half. However, in some women, and it is more common in a first baby, this dilation process can take hours, if not days. You can feel minor contractions of the uterus. They can dilate to 3 centimeters and then stay there for, 30, 36 hours prior to to reaching that next stage in this. So this is by far the most variable of these stages. Often near the end of dilation, the amniotic sac ruptures, releasing the amniotic fluid. This rupture of the amniotic sac can help to facilitate more powerful contractions of the female. So if a female has stalled during dilation, one thing they may actually do is go in and manually rupture the amniotic sac to encourage the development of the process. However, once the amniotic sac is ruptured, the baby is then exposed to the outside world. So if they manually disrupt, rupture your amniotic sac, you're staying in the hospital at that point. You don't get to go home anymore because they want to be able to watch and monitor that to make sure that everything is okay. Ultimately, once it dilates to the massive size of 10 centimeters and is properly effaced, then the contractions of the cervix become more and more powerful, and mom can facilitate the process by contractions of abdominal muscles to allow for the expulsion of the baby to take place. Again, it passes through the cervix and the vaginal canal. The normal delivery is both head first and face down. If the baby isn't in a head first, face down position, what do they call that condition?
Is it breached?
Yeah, breached. The baby is breached, absolutely. They will sometimes try to manipulate the baby inside the womb to try to change the position of the baby. However, typically that will be doing a surgical suite, because if trying to move the baby can put stress on the baby and stress on the mom. And as was mentioned by Maximilian, if there is constriction or increased blood pressure, that can affect oxygen flow to the baby. So it'll be monitored very closely. And if there's any abnormal stress on the baby from that process, then they'll deliver cesarean right there. Is it possible to deliver a baby in the breech position?
Yes.
Yeah, absolutely. Absolutely, it is possible. Is it more challenging? Yes. Is it more likely that maybe the baby can, have a problem, a dislocation of an arm or something like that can occur during the process? Yes. There are more risks to both mom and baby involved in it, but it is perfectly possible to deliver a baby breech, except we live in the world of HMOs. And HMOs are not willing to take the insurance risk associated with those increased challenges with a breech. So typically, if a baby is in a breech position, like I said, they will try to manually manipulate it into the appropriate position. And if it doesn't reach the appropriate position, then they will just deliver it cesarean. And again, not because they can't be delivered breech, but because there's increased risks, and HMOs are risk averse. All right. Again, once baby's out, right, you're checking the color, you're checking to see that it's dry, you're checking to see whether it has a pee-pee or not. right? You're doing all those things. Everybody's happy, sad, excited, tired, everything that's going on. But the process is not complete. As we mentioned, the placenta still needs to be delivered as well. Often, because of all the excitement associated with the baby, placental delivery takes place without even mom or anybody else really, other than, you know, the doctor who's doing it, being aware of it. As I mentioned, when the placenta is delivered, it is inspected thoroughly to make sure that it is completely intact. And in some cases, they will actually even scrape the uterine wall if they're uncertain, to make sure that all of the glandular cells have been removed so that it doesn't interfere with mom's recovery and milk production. As we mentioned and talked about earlier, some societies will ingest it, some will, you know, bury it with a tree, some will donate it to science, because again, in that cord, in that cord blood, there are some of those omnipotent stem cells that we talked about. So a lot of stem cell research that is currently going on is by strains that come from umbilical cord blood. And some people will actually freeze their umbilical cord blood, figuring that, you know, by the time the kid is 30, you know, and they've shot their liver, we may be at a place medically where they can take those omnipotent stem cells and grow them a new liver. And since it's their original cells, there's no chance of the body rejecting it, and so you can get that brand new liver. So, like I said, so it's valuable for research, valuable for you know, some people store it for the potential of it, and like I said, for some cultures or religions, there will be ceremonies associated with the placenta. All right, questions on that? All right, we've gotten baby out, and then there's really just one more thing we need to talk about. Again, we cannot not talk about the female reproductive system without talking about the mammary glands. As we talked about, the breasts in a male and in a female who has never been pregnant is almost exclusively adipose tissue, areolar and adipose tissue. However, with the thanks to that human chorionic somatomammotropin, those immature glands will mature into much more elaborate glandular structures. And their job is, of course, to provide milk. In fact, I think I didn't bring it up, but let me see if I've got it. I thought I had it in the histology. I don't. I bet that Yale site has it. All right, hold on. So in the study tools, Yale histology. There we go. Female reproductive system. Yeah, let's bring you guys back in so you can follow along. So Yale reproductive system, female reproductive system. Come down to the slides. And we want to look at really? Huh. Well, guess we don't have it here. All right, well, I'm sure you can find them online, but again, obviously it's not on your histology list. You're not responsible for it, but the changes in the growth of the glands when they go from immature to a mature state is impressive to look at, but I didn't prepare ahead of time. And since it's not going to be something you're going to need to know histologically, we won't worry about it. Their function obviously is to produce milk to provide sustenance to the offspring. As we already mentioned, again, prolactin is the hormone that produces the milk, and oxytocin is the one that is going to let it down. So as you notice, if you look at the anatomy here, we can see that we have the glands themselves. This is where we're going to produce the milk. The milk is going to swell in these glands. Then oxytocin is going to cause the milk to come down into these enlarged structures right next to the nipple that are the lactiferous sinuses. These lactiferous sinuses basically store the milk so that as the baby sucks on the nipple, it is expressed. So it is that letdown of the milk that is caused by the oxytocin. Notice the adipose end glands themselves are maintained and kept in place by the suspensory ligaments that extend out and attach to the muscular wall. For finishing off the anatomy, as I mentioned, the nipple is where the openings to all of these lactiferous sinuses are located. And then we have the dark pigmented areola that typically become more pigmented upon pregnancy. And what is the purpose of those? What is the purpose of the areola?
Provides a target for the baby.
Yeah, again, a baby's vision is not great, so that provides a high contrast target that makes it easier to find the nipple to express the milk. Absolutely. So we did that, we did that, we did that, we did that. All right, questions on any of this anatomy? All right, excellent. The glands themselves are a combination of tubulo-alveolar glands. They, again, only develop upon pregnancy. They're compartmentalized into individual lobes that have individual lobules separated by connective tissue within them. And again, you're not going to be responsible for that histologically, but in an illustration or a model like this, we have to see that that lactiferous duct comes out of the lobes into that lactiferous sinus, as we mentioned, for expression. Again, the human chorionic somatomammatropin causes the mammary glands to mature, typically by the 6th month of pregnancy, but milk production typically doesn't start till after birth. And even after birth, lactation doesn't truly start right away. Do women produce milk for those first couple of days after birth? No, of course not. We did the hormones already. Instead, they produce this substance known as colostrum. Colostrum for the first couple of days is very, very different from milk. And for starters, it is very, very protein rich and also very low in fat. Females produce typically a relatively small amount of it. And really, it's not necessary for the first couple of days, first day in particular, the baby doesn't require a tremendous amount of nutrients because it just came off of being fully sated by the placenta. So it may take a day or two before the first hunger really hits the baby. And in fact, it is not uncommon for the baby to lose weight from birth to day 1 because of that lack of eating. So again, it's not uncommon as long as it's not a massive amount. That's not a problem. However, after a couple days, the glands convert to milk production. I will say right now that we know that some use the merocrine mode of secretion, which of course, as you know, is exocytosis. It's the most common method used by the body. However, there are some that believe that some of the glands may use the epocrine mode of secretion. Remember, epocrine is where the apical surface of the cell pinches off. This has not been confirmed, so it isn't, we're not going to jump the gun on saying that it is until we know for certain. But one of the things we do know is that typically when the milk is produced, there are really two kinds of milk that is produced. There is what is known as the foremilk. This foremilk is the first milk produced. And the first milk produced is typically more watery and more sugary in nature. Then as the breast is continued to be drained, there is, oops, I spelled that wrong, foremilk. There is the hind milk. This hind milk, which is produced later near the end of the emptying of the breast, is contains more proteins and contains more fat. This is one of the reasons why when breastfeeding, it is typically important to make sure that the baby completely drains the breast before you switch to the second breast. So if you start on the right, try to get the baby to completely drain the right breast and then switch it to the left. And if the baby doesn't completely drain the left breast, then the next time you feed, it is better to start with the left, let the baby completely drain that one, and then alternate to the right. What can happen is that as the breast milk goes from that first milk to that hind milk, it's a little harder for the baby to be able to get the hind milk to express, to get it out. It's harder. And babies are lazy. They don't want to do the work. And the foremilk is very watery and very sweet, and they really like it. So they may get fussy with the breast and want to switch. And the problem with that is that they're not necessarily getting all of the fats and all of the proteins that they need. And just like you, if you eat a real sugary meal or a very carbohydrate rich meal, what happens to you an hour or two later? So nobody here has ever eaten a very carb rich meal before. True, some of it could be that you're tired, but does that sate you for a long period of time? If you have 3 pieces of bread for lunch, are you good till, or even 5 pieces of bread? You have 5 pieces of bread for lunch. Are you going to be good till dinner time? No, you're going to get hungry again. Even though you may have eaten a lot of carbohydrates, they can run through you very quickly. Your insulin spikes, and when your insulin spikes, it packs all that glucose into your cells, and glucose levels drop, and you're hungry again. And the same thing happens with the baby. If the baby eats and then an hour later is fussy again, it may be because they're not getting enough of that hind milk. They're just getting the foremilk, the sugary milk, and they're not getting sated properly. So again, it is important to make sure that the breasts are drained and get that hind milk because it's where the good stuff is. What's also really cool about the mammary glands is that it also it's kind of similar to a positive feedback process that we talked about before. The more you feed, the more the breast will produce. So this is how a mom can feed a single baby, or she can feed twins, or she can feed triplets. Or if you think in medieval times, you could have a wet nurse, a woman in the town who has had a baby, and then the Duchess has a baby, and the Duchess is busy, cutting off people's heads or doing whatever it is that Duchesses do, can't be bothered to breastfeed her own baby. So she finds some woman in the village who happens to have a baby at the same time, and she becomes a wet nurse and feeds both the Duchess's baby and her own baby. So again, it can be based on milk. Now, does that mean that you can, produce enough milk for a whole litter of, babies, right? That Kate and whatever plus 8 or whatever that show was, right? Yeah, you're not going to be able to produce enough 8 just because you don't have the volume in the breast to be able to hold that much milk. But it is basically produced based on use. This can become a challenge when women go back to work, because if they're not able to breastfeed as often, milk production can go down. They do have pumps that can be used to express the milk, and that can help to maintain milk production. But typically it is not as effective at draining the breast fully the way that a baby is able to. And so it can affect the ability to be able to continue to maintain milk production. And again, I think we talked about this, but again, it's another reflex arc. The stimulus is the baby sucking on the nipple. That stretch receptor sends a signal to the hypothalamus, and the hypothalamus neurally controls the posterior pituitary to release oxytocin, just like we saw in labor. And oxytocin causes the milk to let down. Baby is then able to drink the milk, continues to suckle until the breast is drained. Now, as we know, reflexes can be learned reflexes. And often when a baby is hungry, a baby will cry and then mom feeds it. So the problem with that is mom can begin to associate a baby crying with milk letdown. So grandma finally is allowed to take care of the baby for a night so mom and dad can go out and have a nice dinner. And as you're wandering around the mall, maybe doing your Christmas shopping, someone else's baby cries, and what happens?
You leak.
Yeah, absolutely. They can actually express milk as a result of that, the learned behavior that they have. Absolutely. All right. Notice one other thing about this as well. As I mentioned, this is very similar to the reflex we saw with the uterus that causes uterine contractions. And that is another important point. Breastfeeding obviously is helpful for the baby in that it provides food for the baby. It also provides skin-to-skin contact with mom and baby, which helps in bonding. And the release of oxytocin doesn't just cause the milk to let down in the breast for the female, but it continues to cause uterine contractions. Remember, a uterine in a woman who's never been pregnant is the size and shape of a pear. By the time you've reached week 40, it's no longer pear size anymore. So part of the recovery of the female, getting that uterus close, it's never going to get back down to its original size, but getting it close to its original size, these oxytocin contractions can help in the recovery for mom as well. So breastfeeding is helpful for both mom and for baby. And lastly, as we also know, it passes the IgA immunoglobulins on to baby, part of that naturally occurring passive immunity. All right, questions on that? Just that easily, you have reached everything you need to know about anatomy and physiology. We are done. All right, let's go ahead and take one last break. It's 2:23, so we will be coming back at 2:38. And at 2:38, we will do our Q&A review. So if anybody has any questions, and again, this will be for both the lab, well, actually for both the lab and lecture exams and also for the final exam. So any questions you have on the processes, the content, anything else, what you should do when you grow up, you know, what I'm going to do for Christmas, any questions you have, you are welcome and encouraged to ask them when we come back from the break. All right. See you guys in 15 minutes. All right. Before I leave it in your guys' hands, I wanted to show you one more thing. I went to the Cosumnes River College Virtual Anatomy Lab site, and there they actually had some great histology pictures showing the difference between an immature mammary gland, and that is basically what we're looking at here. We can see some of the ducts that are going to be present in their relative size. We can see a large amount of adipose that we can see here. And all of these little things here, these are those immature mammary glands. So we can see some of those structures here. we see a large duct in an active gland, but really what I wanted you to see is this. This is what those mammary glands look like when they are mature. Notice there's these big, huge, massive fluid-filled spaces. Again, notice we can also see the connective tissue that is separating them into individual lobules. So we can see some of the connective tissue septums, dividing them into individual compartmentalized lobules within this whole big lobe. But you can see how much more elaborate and large and glandular, how obviously different they are in their mature versus their immature state. All right, that was the last new piece of information. So as I mentioned, now the floor is yours. This is your opportunity to ask me questions to help you to be successful on all of the remaining exams. So lead on.
Professor, it's fine. You can answer Ruby's question first.
Well, hers is a long one. What's your question? Let's see what your question is, and then we'll go from there.
Okay. You don't have to like draw it out, but if I could just get a quick explanation of how come in meiosis and then meiosis 2, chromosome wise, why are you left with haploid cells each time?
So, okay. Easy. What you have to remember, and again, we won't bother drawing it out, we'll just write it in words. At the beginning of meiosis 1, we have one cell with 46 chromosomes.
Mm-hmm.
All right. And again, at the beginning of meiosis 1, these chromosomes are replicated. Right, we have 2 copies that are held together by that centomere. Okay, that sound great? And remember, this is also the beginning of mitosis. Right, this is how mitosis begins as well. Right, so really at that part, it's no different. What's different is that at the end of meiosis, we have, or end of meiosis 1, let's be more specific again. We have 2 cells. And each of these cells have 23 chromosomes. Right?
Mm-hmm.
1 copy of each unique chromosome. However, those 23 chromosomes that each cell have are still replicated. So at this point, we have 2 haploid cells, but their chromosomes are still replicated. So that at the end of meiosis 2, we now have 4 cells, each with 23 chromosomes, but no, those chromosomes are not replicated.
Got it. Okay.
Yeah, and so that's how we go from the numbers that we have.
Thank you.
Yep. All right, so that answers that question. Ruby, let's go through your question then of the ovarian cycle. The ovarian cycle basically has 2 main phases. And those 2 main phases are the follicular phase and the luteal phase. And what's happening during the follicular phase?
The follicle is growing and maturing.
Excellent. So then what's happening during the luteal phase?
The oocyte finishes meiosis 1, gets stuck in meiosis 2, and then gets ovulated.
Okay, excellent. So you've got the right idea. We need something that is going to trigger the change between the follicular phase and the luteal phase. And that trigger is ovulation. However, remember, our goal in the ovarian cycle isn't so much to talk about what's going on in the egg, but more to talk about what's going on in the ovaries. So you're right, those events are occurring with the egg. That egg has to be ovulated, but after the egg is ovulated, it's no longer in the ovary anymore. So we kind of don't care about it anymore. For the ovarian cycle, we're caring about what's happening in the ovaries. And while the egg has been released, many of those follicle cells remain. But what happens to those follicle cells?
They get converted into the corpus luteum.
So during the luteal phase, we form the corpus luteum, it expands. And does it expand forever?
No, it deflates.
Yeah, it degenerates, deflates, I like that. And goes away. And once it's gone away entirely, we're back into the follicular phase again. So when we're talking about the ovarian cycle, yes, it is absolutely related to what's going on in the egg, but really what we're talking about, and the two names of the two phases tell us what's going on. We have a follicular phase where we care about what's happening with the follicle. We have a luteal phase where we care what's going on with the remaining follicle cells that are becoming the corpus luteum. I like drawing these things because I think it helps sometimes, but sometimes the drawings can be a little confusing. So let's go through the follicular phase. And since you're not going to be able to draw it on the exam anyway, let's go through the different phases that the follicle goes through during the follicular phase. And let's start right on the back. How many phases are there? How many different types of follicles are there?
Four.
Yeah. So essentially there's 4 steps, or we could think of it as 4 types of follicles. What's the first type of follicle?
Primordial.
Primordial.
Oops. Oops. You'd think after 2 years of typing, I would have eventually gotten good at doing this online, but apparently not. All right, we got the primordial follicle. Excellent. Describe what a primordial follicle is to me, for me.
It's a primary oocyte stuck in prophase 1, surrounded by simple squamous cells, or follicular cells, I think they're called.
Yeah. Excellent. So our primordial follicle is a primary oocyte frozen in prophase 1, surrounded by a single layer of squamous cells that are called follicular cells. And we can go ahead and put that in parentheses. Excellent. What's the second type of follicle?
Primary follicle.
Excellent. Describe that.
The oocyte is the same.
So let's cheat. Primary oocyte frozen in prophase 1. Well, here, I'll put the number. There you go. Totally cheat, but that saves me from having to write it out. But what is different is what it's surrounded by. What's it surrounded by now?
Cuboidal granulosa cells.
Yeah, a single layer of cuboidal cells. And they are, again, called granulosa cells. Excellent. What's the next type of follicle?
Secondary. And this also has the same oocyte, but this time it's surrounded by like 2 more layers of cuboidal cells.
All right. Excellent. And then what's the last one?
Tertiary.
And again, we could also call it mature. We could also call it Graphian. Both of those are perfect ways of describing this tertiary follicle. And how would you describe a tertiary follicle?
Secondary oocyte, frozen in metaphase 2. Now with like an antrum and a corona radiata around the zona pellucida.
Well, okay, so I like where you're going with this. It is surrounded by many layers of granulosa cells. And a fluid-filled antrum, which is definitely how we distinguish it. However, be careful. Things like the, oops, no, I want that to stay black. Things like the zona pellucidum, oops, why did that, all right. The fecal cells, those start to appear as early as the primary, right? So these start to appear as early as the primary and continue down. But you are correct. Here, we do have some of those follicular cells that are just around the egg. And those we did call the corona radiata. Again, you don't get a corona radiata until you have that fluid-filled antrum. So absolutely, you are correct that way. And then it is this egg, this corona radiata, that is then released via ovulation. Our secondary oocyte frozen in metaphase 2 is released. And that then leads to our, oh, and what triggers ovulation?
Spike of luteinizing hormone.
Yeah. So there's a huge spike of luteinizing hormone. That luteinizing hormone also then leads to the luteal phase. And the luteal phase really kind of only has 2 phases. I guess we can maybe argue 3. Let's make it 3 phases just for simplicity, or 3 steps. Step 1 is when the corpus luteum grows. All right. Then step 2 is when the corpus luteum starts to degenerate. until finally the corpus luteum goes away entirely. And when it goes away entirely, we're left with a little piece of scar tissue. And what do we call that little piece of scar tissue?
Corpus albicans.
There you go. So we have a story to tell, and these are all the players, all the pieces, all the places in our story for our ovarian cycle. All right? Does this part make sense? Okay, excellent. So then let's work our way through this. We start with dozens of primordial follicles that start to mature to become primary follicles. Is that a fast process?
No.
No. It can take, in some cases, over a year for a primordial follicle to reach the primary stage. And what drives this development? What causes primordial follicles to start to become primary follicles?
Follicle stimulating hormone.
Exactly, follicle stimulating hormone. Perfect. Once we start having primary follicles, they start making estrogens. Right? And that follicle stimulating hormone continues to mature them towards the secondary follicle stage. However, now that we're producing estrogen, are the dozens of cells that may have become primary follicles all going to be able to become secondary follicles?
No, they have to compete for the FSH.
Right.
That's left.
Remember, the estrogen causes the anterior pituitary to stop releasing luteinizing hormone and follicle stimulating hormone, but not making it. So it still makes it, it just can't let it go. And so you're right, less follicle stimulating hormone is present, so they have to compete. All right, until there is a sole survivor. And that sole survivor is strong enough, sensitive enough, gets enough of that follicle stimulating hormone where it matures to that tertiary stage. One winner. And that one winner is huge. So big that this big fluid-filled antrum has formed in there. Also so big that it's producing a massive amount of estrogens. And whereas estrogen was inhibiting the release of the hormones before, when estrogen levels reach really high, they trigger the release. And we get that huge spike of luteinizing hormone that we've talked about. Yes, we get a spike of follicle stimulating hormone, but notice follicle stimulating hormone really helps is this follicular phase is controlled by follicle stimulating hormone, whereas the luteal phase is controlled by luteinizing hormone. So what we care about at ovulation is that huge spike of luteinizing hormone. That huge spike of luteinizing hormone triggers ovulation and converts all those leftover granulosa cells, that burst balloon that we saw, into the corpus luteum. And the big difference between this corpus luteum and these granulosa cells is whereas the granulosa cells produce estrogen only, our corpus luteum produces progestins and estrogens. And those progestins, remember, are the warning bells telling the body to get ready for pregnancy. The problem is these two together inhibit follicle stimulating hormone and luteinizing hormone. So with less luteinizing hormone, we can't maintain the corpus luteum, and the corpus luteum starts to go away, and estrogen and progesterone levels drop until the corpus luteum goes away entirely. And now with essentially little to no progestins and estrogens, there's no more inhibition. And now our primordial follicles can start to mature into primary, and the process continues. Does that help to make sense of it, Ruby? Yep. Again, like I said, I think I'm a visual learner, so I like doing the drawings. I'm a tactile learner, so I like doing the drawings. But I know that sometimes those things get in the way. And I think absolutely taking the time to see all the different stages and define the stages first, and then worry about how we get from stage 1 to stage 2 to stage 3 might make it easier as well. And it might make it easier when you're writing out this as an essay question. You can do this the exact same way. Write out all the stages and then tell me how you get from the one stage to the next stage, to the next stage, to the next stage. That's a perfectly acceptable way to do this. Excellent. Any questions on this process? All right, then I am ready for the next question. Histology of the uterus. Okay, let's see what we got. What? I haven't looked at it, but let's see if it's any good here. Oops. Uterus. Uterus. So, again, with our uterus, there are two main stages. The proliferative stage, where again it is growing back, and there is, or the pre-ovulatory, or there's the secretory stage where it is becoming big and huge, thick and glandularized, producing those secretions, getting ready for pregnancy. I would argue that this is not the best picture. Again, I'm looking at these cold, just like you. What we can clearly see is the division between the muscle layer and the myometrium and the endometrium. I would argue that you can't really easily distinguish the functional from the basal layer. We know the basal layer is obviously closer to the myometrium, and typically it has more cells, so it's a little more dense. So this right here is probably a good example of it, but it gets a little lost up here. It's not the easiest to see. However, while this is not an obvious example of a distinction between the basal and the functional layer, what we can see is that this is definitely pre-ovulatory. The entire endometrium is relatively small, and there are very few glands. We see a lot more tissue than we see glands, which is very indicative of that proliferative or pre-ovulatory stage. Notice here, we can still see the lumen. We can't really make out that it's simple columnar on the surface, but we can tell there's some kind of dark tissue up there on the surface. But notice we maybe see the teeniest bit of the myometrium. Maybe that's the myometrium there. Kind of see this darker region that is probably the basal layer. Again, it's not a lot of it, so it's a little hard to tell. But what we can clearly see is that this is the secretory or post-ovulatory phase. Look at the massive number of glands that are found in here. Huge glandularization, much, much more glands, a much larger expansion of this tissue. This is that endometrium when it is ready for implantation, where it is producing that uterine milk that is going to maintain that blastocyst for a couple days while it floats around inside the lumen before that egg finally implants in the wall and grows into a little baby. And it really kind of is that simple to tell these two apart. The size and the number of glands. Notice as we look back, you can see that, you know, obviously we know the glands are coiled and we can see a little bit of the coils there. But for the most part, we see small pieces of the glands, not a lot of coiling. We see some, we can see some of these coiled glands. We got lucky with the cut through that one there and another one here. But here we can clearly see these big, huge coils of these big uterine glands as they're traveling from the basal layer out towards the apical surface for secretion. All right, does that help? Excellent. What else? Mason, I saw your box become green. I was going to say, did you have another question?
Yeah, I was gonna say, I had a quick question on the immune response. When we did our group projects, that'll be on the final exam, right? But just like the big picture stuff.
Yeah, big picture stuff. Again, I'm trying to think of a good example. The obvious example that I always use is, for instance, like with my 430 class, because we were just talking about their final. If you think of it, you know, like remember when we learned the muscular system, you had to learn all the origins and insertions to a muscle, right? And then you needed to know that material. For the final exam, you don't necessarily need to know the specific insertion. You just need to know what bone, right? What bone does the rectus femoris attach, you know, originate from, right? You don't actually have to know the actual bone feature. So it's kind of a little bit more general type of information. In fact, I would say that, so here's the things I would say about the final. We've had 5 fairly distinct sections. So not surprisingly, our exam will be pretty much cut into 5 distinct sections as well. With 100 questions, that means about 20 questions per section. Some sections had more material, some had less. So for instance, our cardiovascular, or maybe the lymphatic and endocrine and immune response might have a little bit more, whereas digestion all by itself might not need a full 20 questions. So maybe there will be 17 on the digestive system and 22 on the cardiovascular and 21 on the, on the immune response and stuff like that. So, I mean, it's not going to be precise 20, but it'll be close to that. No, you know, no section is going to have 10 questions and no section is going to have 40. So it'll be something close to that. And really, 50% of this information should be information that is just aha information that we've used so much in this class in so many different sections and units that it's just straightforward information, right? What happens when you put CO2 and oxygen? What do you get?
Okay. Carbonic acid.
Yeah, you get an acid, right? Carbonic acid is even better, but absolutely you get an acid, right? What is the most common cation found in the outside of the cells?
Sodium.
Yeah, sodium, absolutely. You know, it's information like that, basic information that we've used a lot in this class that should be pretty simple and straightforward. And again, 50% of the exam should basically be aha information like that. Then, you know, 30% you may have to fire a neuron or two. And then, you know, maybe 20% you have to do a little thinking. Yeah, what muscle did the rectus femoris originate from, right? But again, it should be big global concepts, a little bit of detail, a little bit of anatomy. There may be some images, probably not more than 10% of the exam would be my guess. But again, the other thing about this too is remember, I'm not actually making your exams. I have made the test banks that you are going to be pulling from, but I may have 17 questions on the heart, and I may tell the final to grab 5 of them. Do I know which 5 you're going to get? No. So if, you know, if 4 of those 17 questions are pictures, is it possible that 4 of the 5 that you get are going to be pictures? Yeah, it's not likely, but again, that's with the randomness of this process. So I don't like that I don't have as much control over this final. I've tried to parse it out as much as possible to keep as much control as possible, but there is going to be some randomness to this. So I can't speak as much to the exam precisely because I don't have as much control because of the fact that everybody's getting a different exam. Right, in the classroom, everybody gets the same exam. I have 100% control of which questions are on there. I obviously have 100% control in that I made the test banks. It's not some random picture, you know, some random question off of the interweb that's suddenly going to appear. I picked and wrote the questions, but I don't know which ones you're getting. So
Got it.
hopefully it'll be fair. I've made it as fair as I can in this kind of a situation.
So for BIOL430 over the summer, when I took your class, the final actually, a lot of information, a good amount of information was topics we didn't really cover.
Well, so great question. So here's what I would say. You are responsible for all the information we've covered in this class. The problem with that is that when we, for instance, took the exam on the cardiovascular system, not every concept we covered in the cardiovascular system made it on that exam. It's less of an issue here in this because the exams are online. But one of the things that I warn students about when we're taking this in the classroom is that studying your previous exams for the final exam is one of the worst ways that you can study this material, because you're not being tested on just the things that were on your previous exam. You're being tested on everything that we covered. So maybe the cardiac conduction system wasn't an essay question that you got on your exam for the cardiovascular system. Does that mean the cardiac conduction system isn't going to be on the final? No, of course it's going to be on the final. So you're responsible for all the information we've covered in this semester, not just the stuff that was on the test. And that is especially true in this class because since every of you got a different test, some of you got essay questions that others didn't on the cardiovascular system. So, you know, it really is going to be a collection of everything we covered, not just the stuff that was on the exams.
No, yeah, I understand that, but I felt like a good amount was just not covered in lectures or the Physio X. I thought I just, you know, it was a random, you know, it was just my issue, but I was talking to someone else and they felt the same thing. So I found that a little strange.
Again, I have a little less control with this format, but in making the test, I don't put any questions in the banks that I don't feel that we've covered. Again, as I've said from the beginning of this, one of the things that I love about this class is that it's hard. I don't have to be tricky. I've told you guys before that X, Y, and Z is going to be on the exam, right? We've done it for this one. We spent so much time talking about mitosis, meiosis, oogenesis, and spermatogenesis that I guaranteed you that there'd be at least one essay question on that. And I shouldn't even have to say that. We spend so much time on them. You guys should obviously know that that is a big, huge, important concept. And so likely there's going to be at least one question on that on the exam, right? Because I know I can say that because I know you're still going to have to put in the time and effort to study and learn that. So my goal with these is not to be tricky. So I try to cover the material that we've covered. Again, I focus on the things that we've covered, not the things that have been on the exams, but I try not to have information on that they haven't. However, that being said, the one thing that I would remind you about, the one problem with multiple choice questions is almost by definition, they're a little bit tricky. If a multiple choice question was completely straightforward, then the answer would be obvious and there wouldn't be anything to that. So almost by definition, multiple choice questions, you have to read really carefully. If you misread that one word or if you misinterpret some portion of it, they're going to have that trick answer there that's going to catch you. So they trick you there and they've got the trick answer there to catch you. And again, I know people seem to prefer multiple choice questions, but what I love about essay questions is that they're partial credit. Yes, if you know all the information, you're getting all the points, but even if you know some of the information, you're getting some of the points. With a multiple choice question, you could completely master that concept. Heck, your uncle could have discovered the concept. You could have had your picture taken with that concept. But if you misread that one word in the question, you're going to misinterpret it. They're going to have that trick answer, and you're going to get 0 points, even though you've mastered that material. So that's the only other thing that I would warn you about. Take your time. 120 minutes for 100 multiple choice questions is a lot of time. That's more than a minute per question. If I stood here and stared at you for a minute without speaking, that would feel like an eternity to you. So a minute is a tremendous amount of time, and you have more than that per question. So actually what I encourage you to do is the first time, read the question and try to think what the right answer would be. Then read the question a second time. See if you read the question correctly. Is it still asking the thing that I thought it was asking? And then look at the answers to see if the answer you thought it was there was there. Now, again, for not all questions will that work for, right? But it is something that can help to slow you down and help you to not get caught by some of those tricks from a multiple choice question. So that would be the only other thing that I would say, is that you have to be careful with things that way. But no, my goal is not to make this harder than any other test. And typically averages are about similar to what they are for the lecture exams. So that's similar. Oh, and remember also with your final, it counts as an exam by its own. It's a 100-point exam, counts as 100 points towards your score. But remember, it also can be used to replace your lowest lecture score. And I would say typically in a second semester class like this, easily 80% of students are able to use their final to replace the lowest lecture score. So again, for some people, it may be 2 points, but for someone else, especially maybe someone who didn't have me for 430 and maybe their 430 instructor wasn't as vigorous, they really bombed the first test and got a 50 on it, and they get an 85 on the final exam. That is 35 points added back into your score. So doing well on the final can definitely benefit you in long-term for your grade. So there can be a big swing that way. No, I said there are picture questions on the final. I have no way of knowing how many they're going to be because they're randomly selected. But you got the right idea. It'll be maybe an illustration from your textbook or something like that, and with a couple of labels, and it'll be like, what is, the structure identified by A, or something like that. So yeah, there could be some questions like that. Or like the types of illustrations. Actually, they're more like the types of illustrations in your unit reviews. So in the unit reviews, the first part, to check your recall when you're labeling things, those kind of images with a label, and you have to say what something is. And again, at this point, you should be able to still, on a final exam, be able to recognize the inferior vena cava, right? Or the left lobe of the liver, or the cecum of the large intestine, or something like that. Those are things that you should easily be able to do. Or the Incidiotrophoblast from the embryo as it is embedding itself into the wall of the uterus.
Actually, Professor, on that topic, because we weren't given a histology thing on that, would you just give us like a diagram that you would find, like a picture that was drawn to identify like a Incidio or a cytotrophoblast?
So yes, so notice we looked at illustrations for these things today in lecture. I don't give a handout because in this section, I'm not sure always how far I'll be able to get and how much we'll be able to cover, but you are responsible for all the anatomy of the things that we talked about today. However, notice specifically there's no histology. So while we looked at that dividing, I guess that would be the one case. If I showed you a microscope slide of that dividing zygote and I asked you to identify a blastomere, that would pretty much be the only thing that I could see on that. So maybe a blastomere on that, but otherwise all these things that we've done would just be illustrations. So the same kind of illustrations we used in class today, those would be the things that you'd be responsible for.
Thank you.
I don't think, even though we went over the mammary gland, I don't think is in there too. So again, so hold on, wait, I think there were a couple. Yes, it will be 100 multiple choice questions, just like a normal lecture exam. So they'll all be on the same page like that. Because even though there may be a few images, it's not as image rich as the lab exams. So I like that better too, because you can kind of bounce around to the ones that you're comfortable with. Yeah. Yep. Any more questions? All right, I will remind you that I have office hours tomorrow. I have office hours before your exam on Thursday. I'm good at responding to emails. So again, you guys have all those opportunity. That is an excellent question. My goal is to try to get your exams graded and posted prior to the final. However, I can't guarantee that I'm going to be able to do that. My 430 class is taking their exam tomorrow, and so I'll be working on both your exams and their exams at the same time. So I will make every effort to get them done ahead of time, but because you guys have a short turnaround of those 5 days, I don't know if I will be able to. So I will try, and if I do, I will post them, so the grades. And then if I post the grades, you can come to my office to see the exams. I won't release the exams before the final, but you can see it if I get them graded. But I can't guarantee you that I will. I will promise you that I will make every effort. I will make every effort to get them graded ahead of time, so you know where you are in the class and how well you can do in the final. But I can't promise you that I can get it done. So I promise you I will try. But do the best you can on the final, because again, think of it this way: every point you get above your lowest lecture score counts as 2. So the better you do, the more points you're going to get, the more successful you're going to be in the class. Yes, the final, our class starts at 12, and again, you will have the final exam at that point. And like I said, the only difference is you'll only have 2 hours to complete the exam. So other than that, it works exactly the same way as all your other exams. Yep. All right, guys. It has been a pleasure. It's been an interesting semester. I congratulate you guys for surviving anatomy and physiology online. Hopefully you're going on to bigger and better things, and hopefully those will be on campus for you somewhere. I will be on campus. So look at the bright side. If you totally bomb the final and have to take this again, you can take it with me on campus. So think how much more fun that will be. Although I know all of you are hoping to not have to see me again, and I don't take that personally. So I wish you all tremendous success in the rest of your academic careers. I hope you are successful in getting into what it is that you want to. Thank you. Merry Christmas to all of you guys as well. Have a wonderful, wonderful break. Do well on the exams. If you need anything, come to my office hours or email me. Otherwise, take care, and I will see you guys. Have a good one.
Thank you.
Thank you. Have a good holiday.
Yep, you guys too.